Good day, everyone. I'll be discussing an important biochemical test, which is usually one of the first tests that is performed in identifying an unknown organism, the Catalase test. The Catalase test is based on the principle that if catalase is present in a bacterial cell, or if it is produced by a bacterial cell in the presence of hydrogen peroxide, it will catalyze the breakdown of hydrogen peroxide into water and oxygen. The bubbling and frothing that is seen in the positive test reaction is due to the escape of oxygen that is produced. There are two types of catalase tests. You have the slide catalase test and then the tube catalase test. It is important to note that the catalase test detects the enzyme catalase in bacteria. Is also essential for differentiating catalase positive Staphylococcus from catalase negative Streptococcus. The procedure involves emulsifying a colony from a well-isolated 18 to 24-hour culture, and then on a microscope slide. This is followed by addition of 3 drops of hydrogen peroxide. In this plate, showing the slide catalase test result, the upper slide shows the positive reaction produced by Staphylococcus aureus. Notice the bubbling and frothing due to the escape of oxygen, while in the bottom slide, glass slide, is a negative reaction produced by Streptococcus pyogenes. The second type of catalase test is the tube catalase test. Here, 4 to 5 drops of 3% hydrogen peroxide are placed in a test tube. Then a small amount of the organism, the test organism, from a well-isolated 18 to 24-hour colony is picked using a wooden applicator stick, and placed in a tube. It's important to be very careful in picking this isolated colony, because if the colony is picked from an agar containing red blood cells, a false positive reaction may be observed because of the carryover of red blood cells into the test tube. The tube is now placed against a dark background and observed for immediate bubbling and frothing. Here is plate 2 showing the positive tube catalase test. Note or take note of the bubbling of the oxygen that is escaping following the breakdown of hydrogen peroxide by catalase. while in this plate is a negative tube catalase test. There is no observed bubbling or frothing. That is, no oxygen is produced. That means catalase is not present in the bacterial cells. What is the principle behind the catalase test? Catalase is an enzyme which is present in human, plants, and animals, as well as microbial cells, except in Streptococcus species. So it's more significant where it is negative. So the organism to suspect is Streptococcus species. The catalase breaks down hydrogen peroxide to water and oxygen, and the bubbling observed in a positive test is due to the escape of the oxygen produced in the reaction. The application of catalase test, all microbial cells are catalase positive except Streptococcus species. Here are some selected references and links for further reading. Thank you for watching.